Hello students, myself Popere KS, Assistant Professor in Botany at Red Shikshan Sanstha's RBNB College, Srirampur. In this lecture, we are going to discuss the next topic, that is the microbodies. Now, this is also a organelle which is found in cytoplasm of a cell. Now, this microbody, these are the organelle which is So this is an organelle which is bound by a single membrane or single unit membrane. It has presence of only one membrane outside. Now next is in its matrix. It is electron dense. So inside the organelle, that is the microbodies, it is electron dense. Now in matrix, it also has presence of different protein. So it has presence of protein and enzyme in the matrix. Now there are constant there is a constant that is the phospholipid. They are constantly entering into the microbodies, so they are involved in the synthesis of the expanding microbodies or the new microbodies which are forming. So this phospholipid, it contributes to the new forming microbodies or the new which are continuously forming. So this phospholipid, it is continuously transferring into the microbody. So there is a continuous that is the means coming into the microbodies. Now on the basis of this different types of enzymes and the proteins and the type of a cell in which these microbodies are synthesizing, they are classified into 4 different types. So on the basis of the enzyme present and the type of a cell in which the microbodies are occurring, it is divided into So they are classified into peroxisome, glycosomes, hydrogenosomes and sperosomes. So these are the 4 types in which the microbodies are divided, or these are the 4 types of the microbodies. Now we are going to see the functions of each one by one. So first one is peroxisome. This peroxisome, the term, that is the first, it is given by a scientist, D. Dewey. So this was the first scientist who has given the name of the peroxisome in 1969. Now this peroxisome, they are the tiny circular membrane bound organelles. They contain different enzymes. So these are just a spherical like structure, round oval structures which consisting of the different types of the enzymes. Now the enzymes which are present in the peroxisomes are peroxidase, then urate oxidase. So there are many etc. So many different types of enzymes they are present, and they have mostly the enzymes which are present in the microbodies, they have the role in detoxification of a cell. So mostly the enzymes which are present, they have a major role in detoxification of the cell, like the metabolism of the hydrogen peroxide that will be released out of the cell by the different enzymes. Now in case of the plant, in their leaves, this peroxisome, their photosynthesis will occur. So this is about the peroxisome. Now we will see the morphology of peroxisome. Now we have already discussed, it is mostly round oval. So they are mostly round, oval or small bodies that may be spherical. The number varies from 70 to 100 in a per cell. So in a cell there might be presence of 70 to 100 peroxisome in a cell. Now the size is from the size it might be from 0.2 to 1 mu in a cell. Now it also has presence of a single membrane. So it is also bound by a single membrane that may be of 60 angstrom in thickness. So it has presence of only one membrane, peroxisome, and that will be across 60 angstrom. Now these membranes, they are selectively permeable. So it will be permeable to some selected amino acid like hydroxy acid and uric acid. So they are permeable, or we can say selectively permeable. That is to certain amino acid. It is now, so it will allow some acids and amino acid to that pass through the membrane of the its membrane, but there are some we can say nucleic acid or pyrimidines, that is cytosine and thymine, that are not allowed to pass through the membrane of the peroxisome. So this cytosine and thymine, that it is in pyrimidine which is not allowed to pass through the membrane of a peroxisome. Now we will see the chemistry of this peroxisome. So as we have already discussed, it will consisting of the different enzymes, and the enzymes which have role in detoxification of a cell. So firstly they will contain it consists of a glycolytic enzyme. Now what this glycolytic enzyme do? It will involved in the breakdown of the glycolate. So glycolate breakdown is done by the glycolytic enzyme. Now the other enzymes which are present are peroxidase. Now this peroxidase, it has role in metabolism of hydrogen peroxide, that is H2O2. Next is D-amino oxidase. Now it also has role in the, that is the detoxification, that is in the metabolism of hydrogen peroxide. Next is next is urate oxidase, and one more is there, that is catalase. So this is involved in metabolism of H2O2, and the enzyme, that is D-amino oxidase and urate oxidase, they are involved in production of H2O2 in the cell. So these two are related to, means all have mostly the function related to hydrogen peroxide, that is the metabolism of H2O2, but these are involved in the metabolism of H2O2. Now these two are involved in production of H2O2 in the cell, and this catalase, it oxidizes and decomposes the hydrogen peroxide to remove the toxic effect in the cell. So it is involved in the oxidation and the decomposition of H2O2, and it will remove the adverse effect, or we can say the harmful effect of the H2O2 from the cell. Now we will see the functions. Now this peroxisome, they are the most important organelles. These are, in case of the plant, mostly they are involved with the synthesis of amino acid, so in case of the plant, they have function in the synthesis of the various amino acid. They are involved in the synthesis in case of the plant mostly. Now peroxisome, they are also associated with the photorespiration in case of the C3 plant. So in photorespiration, three different organelles are involved, that is the peroxisome, mitochondria, and the the three organelles are involved: peroxisome, mitochondria, and the chlorophyll containing compound, that is the chloroplast, is involved. So this three organelle of them, one is most important, that is the peroxisome. So these are involved in the C3 cycle, that is C3 plant, for the photorespiration. Now these are also involved in, as we have seen, there are different enzymes present. So these are involved in that is the adverse effect So it deals with the adverse effect of the H2O2, that is the hydrogen peroxide. Now one more important thing is there, that is so this is the most important step, that is the beta oxidation of the fats. Now this process, it also occur in the peroxisome, that is the beta oxidation of the fats. Now next organelle, now this is about peroxisome. We will see glycosome, hydrogenosomes, and spirosomes also in short. So first is glyoxysomes. Now this glycosome, it is also a type of microbody. This will be mostly seen in the plant, or we can say germinating plant, or in the seed. So they are seen in the germinating plant or the seed, and in their matrix it will be consisting of, in the matrix of the glycosome there is presence of amorphous protein that will be present in the matrix, and it will have a unit membrane, that is a single membrane. The membranes of the glycosome, this is formed by the endoplasmic reticulum, and the enzymes of this glycosome, they are synthesized on the free ribosome. This microbody, that is the glycosome, as it is mostly present in case of the seed or in the germinating plant. So what it will do, it will convert the fat or the oils which are present in the seed into sugars. So there will be So it will be converted, the fat or the oil which are present in the seed, it will be converted into sugar. Now this is the usable form of the energy, or now the sugar, it can be used for the utilization of the energy. Now they also help in the breakdown of the ethyl alcohol. So the breakdown of the ethyl alcohol, it will take place in glycosome. In this, hydrogen peroxide is released as a byproduct when itself broken down into H2O and O2. Now next is hydrogenosomes. Now this hydrogenosomes, they are also microbodies. It is a type of microbody. These are the organelles which are double membrane. So as we have seen, the peroxisome, glycosome and even the sperosome, they will be single membrane or they have a unit membrane. But in case of the hydrosome, it is a type of the microbody which is double membrane, and they produce So production of the hydrogen, it will take place in glycosome extensively. Now it also, one peculiar feature is that it has a presence of genome. So genome is present only in case of the type of microbody, that is the glycosome. It lacks the enzymes which are involved in the TCA cycle. So lack enzymes of TCA cycle. So it don't have the enzymes which are involved in the TCA cycle or citric acid cycle. They also now this hydrogenosomes, they also has originated from the mitochondria when there will be losing of the respiratory enzyme. From the mitochondria, when the respiratory enzyme it is loses, then it gets converted into the microbody, that is the hydrogenosomes. Now last one, that is the sperosomes. This peroxisome, now this is the last type of the microbody. Now this also have a presence of single unit membrane. So only hydrogenosome, they are double membrane or they have the presence of double membrane. Now this will be this sperosomes, they have presence of single membrane. Now this sperosomes, they will be produced in a seed that will have alluron layer. So they occur in abundance in the alluron layers. They synthesize and store fat. So this will be mostly produced in the alluron layer and they synthesize and store fat. So this is about the sperosome. This is all about that is the microbody. So this microbody, there are 4 types, that is peroxisome, glycosomes, hydrogenosome, sperosome. So all will have some different function. Of this, the peroxisome, hydrosomes and sperosomes, they are having a single unit membrane, but this peroxisome, glycosome and sperosome, they have single unit membrane, but the hydrogenosomes, it will have presence of double membrane, and it also has a presence of genome. So this is all about microbodies. Thank you.